Okay, so we're doing the biology paper 1.1, the first test. The questions are on this side, and I've got the main information there. It says, students investigate the activity of enzyme lipase at different temperatures. So I've got the same amount of lipase, 1 centimeter, and I've got the same amount of milk, just at different temperatures. There it is in a table. I can already see that the enzyme activity is quite low at a low temperature and quite high at a higher temperature. So the first question is choose a scale. So we're going to plot these by choosing a scale first. You get 1 mark for this scale. If I just show you that, it's 1 mark. So the scale is quite easy because you've got enough boxes to go up each small box by 1, so the big box becomes 10, so the increments of 10 there. Plotting results for the enzyme activity shown in the table opposite, so that's worth 2 marks. So what I've done there is just simply plotted these results against, so when it was 10 it was 15, the enzyme activity, so you can see that I've come across there 15. And then it was 32 when it was 20 degrees, so I got 20 and 32, so it's just plotted accurately. And then your last mark then is for joining, that says there. Yeah, joining your plots with a ruler. And that's exactly what I've done there. So I've just joined my plots with a ruler. And then the next bit says, so let me shrink that on there and put that there, because it is quite difficult over 2 pages here. It says, describe how the activity of the enzyme changes between temperatures 25 and 45. So to do that, you just look at 25 there, 45, and it does increase. So it increases. So it rises, and I've said falls there because it's to 45. So 45 there, so it increases and then falls. So rises then falls. Calculate the change in enzyme activity between 15 degrees Celsius and 35 degrees Celsius showing you're working. So it's 2 marks. You get 1 mark for showing you're working and another mark for the answer. So to do that, between 15 and 35, we get this again. We've just drawn the line up. 15 is 22. 35, it's 51. I've simply subtracted 22 from 51 to get my answer there, which was 27, I think. Anywhere between 27 and 29 on the mark scheme. The next question, let me shrink that a bit more. The student set up a controlled test tube using boiled lipase this time. So that's already suggesting that this lipase has been denatured, because that's what happens. If you boil an enzyme, then you're going to denature it, it's not going to work. State the volume of boiled lipase solution and milk that should be used. Well, if we look back at the first page, they used 1 centimeter cubed lipase and 5 centimeter cubed milk. We've got to keep it the same. If we're going to keep it a fair test, it needs to be the same. So I've said 1 and 5 there, and the reason it needs to be controlled. It needs to be a fair test. You need to, you're measuring what effect that the boiled lipase has, not, and if you go changing any of these, then it's not a fair test. Give a reason why there was no enzyme activity. Well, like we say, because it's boiled, the enzyme was denatured, so it's not gonna have any effect. And then question 2, the diagram below shows an algal cell, so that's algae. So a little bit naughty here, because you might not have come across this, but so if you can recognise that's pointing, if it's pointing at nothing, then it's likely to be cytoplasm. The cell wall is the outer one, and the cell membrane is the inner one. And then you've got the nucleus and large chloroplast. Complete the table. So the nucleus is it contains DNA. It controls the activities of the cell. Photosynthesis occurs in the chloroplast, and then the cell wall provides support and strength. It says, name one part of this cell shown in the diagram which is not present in an animal cell. So it's got to be in this cell. And I've said it's either the chloroplast or the cell wall. Okay, you can't have vacuole because there's no vacuole there. And then name one part of the algal cell shown in the diagram above which is not present in a bacterial cell. So there's no nucleus in a bacterial cell, and there's no chloroplast in a bacterial cell. Question 3. Complete the sentences using some of the words below. So, osmosis, water moves from an area of high concentration to an area of low concentration through a semi-permeable membrane. So there's 2 marks there. Just like diffusion, high to low, only osmosis is through a semi-permeable membrane. And then state the number of the tube which contain the lowest concentration of water. So the lowest concentration of water means that it's going to contain more of the blackcurrant squash. So you can see that's darker, strong solution of blackcurrant squash. So that is the lowest concentration of water. The highest concentration of water is this one because it contains more water. I think we may need that, so I'll just keep it out a sec. After 30 minutes, they removed the potato chip and recorded the mass of each. So, before we do the table, we come down, use the bar chart to complete the results table above. So, you can see that this one's been done for us. So, if I look at the keys, the dark is the mass at the start, the light is the mass after 30 minutes. Now the mass at the start, the 3 has been given to us and they're all 20. So I've come down and given all 20 there. So this one's 20, this one's 20, and this one's 20. This one's also been given to us. It's 24. A lot of students make a mistake, they say 23 and think they go up in ones, but because that's 20 and 20 and that's 25, every 2 squares is worth 1. So we've got to be careful there. State the number of the tube in which the concentration of water in the chip was the same as that as the solution. Well, that's evident there, it's number 2, because there's no change in mass. And then explain why the potato chip in 1 has gained mass. So it's because the water concentration was higher outside the chip for 1 mark. So water is absorbed by the chip via osmosis for your next mark. And then the last question, which is a 6 marker. Let me zoom in. So an experiment was set up, Viscin tube as a model of a gut. So this is the Viscin tube. This is shown in the following diagram. The Viscin tubing was filled with starch solution and 1% amylase enzyme. So already I can link the enzyme with the starch. I know that the enzyme amylase breaks down starch into glucose. After 30 minutes, the water surrounding the Viscin tubing was tested, and it contained glucose. So this water contained glucose but no starch. Explain why glucose appeared in the water but no starch was found, including in your account the description of how the water was tested using Benedict's and iodine solution and your expected observations. So there's a lot to work for 6 marks there. So the first thing we've got to recognize is the Viscin tubing is a good model for the gut because it is semi-permeable, so it allows some substances in and out. Amylase, as we said, is the enzyme that breaks down starch into glucose. So starch is large molecules where glucose is small enough to pass through this membrane. So starch is going to not be able to get out of this, but because it's only 1% amylase, there's still going to be starch present in here, but because it's breaking it down into glucose, some of the glucose is going to transfer through this membrane and come into this water. So like I've said there, the starch molecules are too big to fit through the Viscin tubing. Glucose is small enough. The glucose will diffuse, and that's the word, it's not osmosis, it's diffuse into the water because it's a higher concentration inside the Viscin tubing. So be careful, it's not osmosis. Osmosis is the transfer of water through a semi-permeable membrane. Benedict's solution can be used to test for glucose. It will turn from blue to orange if it's positive. If it's positive for glucose. If there's no glucose, obviously it will remain blue. And then you can test for starch by using iodine. You should go from that sort of brown colour to a dark blue-black. Again, if it's a positive result. And that's your marks for this question.